Welcome to The Explainer. Today, we're decoding the master blueprint that governs the entire microscopic world. We're talking about bacteria and viruses, whose genetic secrets can literally be a matter of life and death for us.
It's a huge question in modern medicine, right? And the answer isn't some kind of magic, it's genetics. You see, microbes have this absolutely ingenious ability to rewrite, edit, and even share their own genetic code at lightning speed. So let's break down exactly how they do it.
All right, so to really get how microbes pull off these amazing genetic tricks, we have to start with the blueprint itself, their DNA. It's the fundamental molecule that holds all the instructions. Okay, so think of the microbe's entire DNA like a vast architectural plan. A single gene, then, is like one specific instruction on that plan. Something like build a support beam or install a window. Each one dictates a very specific part of the cell's structure or a function it needs to perform.
And this brings us to a really crucial distinction: genotype versus phenotype. The genotype is the entire set of blueprints, all the genetic potential an organism has. But the phenotype is what you actually see. It's the finished building, the traits that are expressed, like a bacterium's color or, and this is a big one, its ability to survive an antibiotic.
So the cell has the blueprint. How does it actually read it and start building things? Well, this whole process of gene expression happens in two essential steps. It's all about turning information into action.
Here's the first step. It's called transcription. Now, don't get lost in the details here. Just focus on the main idea. You have an enzyme that acts like a super meticulous photocopier. It finds one specific gene on the DNA and makes a temporary portable copy. This copy is called messenger RNA, or mRNA, and it carries the instruction out to the cell's construction site. Now, the genetic language on that mRNA copy is written in these three-letter words called codons, and each three-letter word corresponds to one specific amino acid, which are just the fundamental building blocks of proteins. The whole system is basically a dictionary, translating from the language of nucleotides into the language of proteins.
And that brings us to step 2, translation. A structure called the ribosome acts as the protein factory. It clamps right onto that mRNA copy, reads the three-letter codons one by one, and assembles the right amino acids into a chain, creating the final, functional protein. Simple as that.
Now, this process is incredibly precise, but, it's not foolproof. So what happens if there's a typo in the master blueprint? A permanent change in the DNA sequence? Well, that's what we call a mutation. It can be as simple as a single typo, one incorrect base put in place while the DNA is being copied. But that small mistake gets locked in and passed down to all future generations, permanently altering the blueprint from then on. And these typos can have wildly different outcomes. A missense mutation swaps one amino acid for another. Could be a minor tweak, could be a major problem. A nonsense mutation puts a stop sign right in the middle of the instructions, which just leads to a short, useless protein. But a frame shift mutation, whoa. That's where you add or delete a base, and it scrambles the entire message from that point on, usually resulting in a completely non-functional product. But here's where microbes get really interesting. See, they don't just sit around waiting for random mutations to change their own blueprints. No, they are absolute masters at sharing plans and genetic information with each other. We're all familiar with vertical gene transfer. That's just parents passing genes to their children. But bacteria specialize in something called horizontal gene transfer. They can pass genes directly to their neighbors and their peers, even if they're a different species. It's just like an architect handing over a new, improved blueprint to a colleague right in the middle of a project. It's a real game changer. And they've got 3 main ways of sharing these genetic secrets. Each one is a fascinating mechanism that allows for incredibly rapid evolution. Let's take a look at them one by one. First up is transformation. This is where a bacterium can literally just scavenge for fragments of naked DNA from its environment, usually from other bacteria that have died, and then it just incorporates those genetic plans into its own code. Next is conjugation, which is the closest thing bacteria have to mating. This one requires direct cell-to-cell contact. One bacterium actually extends a physical bridge called a pilus over to another and then directly passes over a copy of a plasmid. That's a small, circular piece of DNA that often carries really useful genes, like the ones for antibiotic resistance. And finally, there's transduction, which cleverly uses a virus as a middleman. A virus that infects bacteria, it's called a bacteriophage, can accidentally package up a piece of its host's DNA.
Then, when that virus goes on to infect a new cell, it injects the previous bacterium's DNA instead of its own, basically acting as a genetic delivery service.
Okay, so we've got blueprint editing through mutation, and we've got blueprint sharing through horizontal gene transfer. So what? Why is this so critically important for us to understand?
Well, these mechanisms are the very engine of microbial evolution. They are behind one of our biggest public health challenges, the shockingly fast spread of antibiotic resistance, where a single resistance gene can just sweep through a whole hospital. But on the flip side, because we understand this genetic toolkit, we've been able to harness it for our own benefit in biotechnology.
And this is a process called genetic engineering. We can use these same microbial tools, like plasmids and DNA-cutting enzymes, to insert a useful gene into a bacterium. This effectively turns that microbe into a tiny living factory, producing valuable proteins for us, like insulin or human growth hormone.
So in the end, microbial genetics is a real double-edged sword. It's the force driving the evolution of dangerous superbugs, but it also gives us these powerful tools of genetic engineering. The secrets written in this constantly changing microbial blueprint are still being unlocked, and they hold the potential for new solutions in medicine, in agriculture, and, well, beyond.